Bueno, ¿qué tal? Dar honor a este tan importante webinar que vamos a presentar.
Hello, it's an honor to bring you this important webinar. We believe that it deserves all the time that it's going to take. It's going to be important to hear from all the experts. So that's why I wanted to be very much on time for the presentation. Once again, we're here. Thanks for being here. So we can share this moment with you. You know that we are working all together, and we're working in order to have our dear society be part of an important moment like this. As you know, throughout time, we've done or put together many meetings and activities in order to be here. And for this, I want to take a moment for the next presentation. That's our call to the next Secretary for LACID. This is a very important position where we try to get a lot of people registered. They're excellent candidates. We have a picture of somebody like Dr. Ekaterini, and also so many people that can be here, because I believe you could be very good secretaries and in the future, president of LACID. So we are waiting in this opportunity. Don't forget the date. So hopefully we're going to have many candidates. We want to tell you that we're going to have interpretation into English and Spanish, so you're going to be able to choose English or Spanish. This is something that we do in order to reach as many of you as possible. We want to celebrate the International Day on June 28th, International Neonatal Screening Day. It's very important for us. It's pioneer in the region, Brazil. We want to congratulate all the team of immunologists that are doing an extraordinary job in immunodeficiencies, and particularly neonatal screening for the early diagnosis of immunodeficiencies. So once again, congratulations to the team of immunologists. Also, I want to tell you that the class on cytometry for the National Diagnosis of Immunodeficiencies, this was by Dr. Isabel Gallard. You can find it in our social media, in our network. You can find this as genetic diagnosis in the syndrome for innate errors in immunity. We had a symposium that was wonderful, and this was headed and coordinated by Dr. Ignacio Uriarte Aratu. It was very good, and also you can find all the conferences that have been recorded in our web pages. We want to call on July 15 to be with us to see what the young immunologists are doing in Latin America. What is the future? What is the area where they can develop? What is their visions for our young people in La CID? And where we can have a discussion place. What can we propose and what they propose to us? This is our next webinar, July 15, 2021, with our dear Alejandro Palma, that is going to be the coordinator. And that's going to be fantastic. We hope that everybody is here with us at our meeting for 2021. It's going to be virtual, led by the entire team of Chilean immunologists that are coordinating. Cecilia Poli, 13 to 16 of October. And I'm sure this is going to be an extraordinary meeting, as our colleagues are used to in Chile. And we're going to try to give you the best in order to be there as society. We need to continue being part of the registry. We need to have access to the registry for our patients. Dr. Gisela Seminario is here, and it's the presentation of our paper on what happens in this region with our patients with the innate errors of immunity as we face COVID. We have a lot of experience, and this should be communicated and it should be shared with the rest of the world. So once again, I call you. This is work that we must do. Next, to be part of our society is so important. This was shown at the pandemic. The pandemic has shown us that we can be together, that we can work together, that we can present, that we can submit our results. And this is so important. So we ask that you continue supporting it, that you invite different friends, different professionals in each and every one of your countries to be members. And as always, we're going to have our meetings. They have this characteristic of having this warmth, and very soon we're going to be able to do it in person. Now, today's idea is to present a view of the different countries like Argentina, Brazil, and the experience about innate errors. As we can see from Australia, we're going to have also Kate Sullivan from the United States. We have also our colleague, like I said, in Australia, Ekaterine, and our professor from the Department of Pediatrics in the School of Medicine at the University of Rio de Janeiro. That is one of the references in allergy immunology in Brazil. She's going to be our coordinator, and I really thank her for being here, to be here with us, as always. So you have the floor, Ekaterine.
Tonight for this webinar, we'll have speakers that are really very well known and dear to all our clinical immunology community, but a little formality won't do no harm, so I'll introduce them properly. And at the end of the presentations, I will help with the questions. First, we'll have Professor Guilhermo Docena, a CONICET principal researcher, professor of immunology at the National University of La Plata, Argentina. Then we'll have Liliana Berzrodnik, the current president, great president of LASID, also a CONICET researcher and director of the Jeffrey Model Argentina Center. Then Antônio Condino Neto, professor of experimental medicine at the University of São Paulo, a great past president of LASID. Then we'll hear Stuart Tanji, researcher at the Garvan Institute of Medical Research, a professor at the Faculty of Medicine at the University of New South Wales, Sydney, Australia. Then the dear Kate Sullivan, chief of the Allergy and Immunology Division at the Children's Hospital of Philadelphia, past president of CIS. And then Anthony Lacava, Allergy and Immunology Fellow at Penn. He works at the Hospital of the University of Pennsylvania. So welcome you all. And Dr. Professor da Siena, please, you have the word.
Buenas noches para muchos, buenas
Good
tardes.
evening for many, good afternoon for others. Really, for me, it's a pleasure to share with you this activity. Along with LASID, is such an important association for all of us. So I want to thank Dr. Liliana Verasnik, who has invited me to be here to share with you some of the results that we have in a project that we have in Argentina, and also to thank the authorities of LASID for allowing me to be here. As I said, I belong to the National University of La Plata. I'm at an institute that depends on CONICET and the university. And last year, at the beginning of the pandemic, they created a multidisciplinary group COVID Exactas, that has been working in different areas that do not have anything to do with so many areas. When they call us from the university to try to support from the areas of investigation, our institute studies basic aspects of immunology. And this is what the group is put together. We have been working all year in the development of diagnosis methods. And currently, we are working in the development of a COVID vaccine. At the beginning of the year, after the vaccination stage started in our country, in the province of Buenos Aires, we started putting together a group of different disciplines. It's not only immunologists, but to study the immune response individuals that were vaccinated in the province of Buenos Aires. Currently, it's a huge pleasure. 6 months after starting the vaccination, to be able to show you this information, these numbers in November, December, when they were just giving the first vaccines. We have 16 vaccines now. I believe that the New York Times, this app, is really interesting because it's an update. It has an update every day. And you really can be surprised, because when I have to make a presentation, I like to see these figures to underscore one of the positive aspects that we have from this pandemic, even with all the health disaster that is living throughout the world, the development of new platforms, and to be able to show the world, society at large, how the scientific community, how everybody is there for the development of vaccines, because we pretty much have 120 vaccines in clinical trials, and I know that they're being given in individuals, 100 more that are in preclinical stage, like ours in Argentina, along with other 5 projects in Argentina and 6 development projects for vaccines in preclinical phase. And currently, we have 16 vaccines that have been authorized that are being used throughout the world. And you can see how this chart, this graph, it's really impressive how day in, day out, the number of doses in the world increases. And we currently, we have about 40% of the population, maybe a little bit more, a little bit over 7 million vaccines that have been distributed in different countries. A problem that we see and that we're facing as particularly the countries in the Southern Hemisphere is that there's inequalities in the distribution of vaccines throughout the planet. And we can see that many countries have percentages of their population really advanced with respect to the vaccines they have received. And on the other half, there are other regions, Africa and some other countries in Latin America, Brazil, Argentina, and Chile, where the percentage of the vaccinated population is a lot less. We can see how we're getting close to what we consider statistically that is going to be herd immunity. So we can see cases like Israel, where 61% of the population has received the vaccine. Pretty much all of them have the complete scheme. It's really interesting to see that Chile, what's happening in Latin America, in England, in the United States. But what is really interesting and that we see is that after 40% or from 40% and up of the vaccinated population, you start seeing changes in the curve of transmission of the disease. After the vaccination, the curves have drastically changed depending on the country, depending on the conditions under which this can accompany the vaccination plan with the voluntary measure plans. That has an impact in the curve, but in November, December, second, third, and fourth transmission waves, and then everybody started vaccinating in the world. We didn't know how the different groups were going to respond and how were they going to be able to change this curve. So there's no question that vaccines are safe, have efficacy and efficiency, and that they can change these contagious curves, and that they're going to be able to change the course of the pandemic. So the vaccination went from being promising to be a huge promise in order to stop and control the pandemic. However, as I mentioned, the limited provision of vaccines in certain regions and countries in the planet has created the situation of designing alternate strategies for the vaccination, and to have even currently, we are considering different strategies where there should be a priority for the groups that are exposed more with those groups that have more risk. Our country is in that situation. These are the numbers in Argentina. We have a 33% of the population that have received the dose, a very low percentage, 2 doses, 18 million vaccines. And in the past few weeks, this kinetics of the arrival of the vaccines have changed, but we still have a long way to go in order to achieve a higher percentage of the population vaccinated, and to try to keep with time what we see in the past 3 weeks in Argentina, which is contention and a reduction in the case in the number of cases infected, in such a way that we can say that these vaccines are working in Argentina, even though it's so difficult to keep restrictions here. In Argentina, we have 3, Polype, Oxford, AstraZeneca, and Sinopharm, but I'm going to be talking more about Sputnik V, because most of the individuals have received it, particularly the first dose, and also because outside of Russia, this is the country that has received the largest number of vaccines of this type. I can say, and everybody knows, what Sputnik V vaccine is. It's a vaccine with a platform based on nucleic acids with 2 viral vectors, non-human because the genes, the early genes have been replaced for the genes that code for S protein. So when this virus that carries the information for the production of this viral antigen goes into the cells of our body, mediated by a receptor in all the cells. Here we see the release of the DNA that codes for this protein and for others. And this is translated, the protein S is translated, is released from the cell, is anchored, is expressed. And what's true is that the availability of the protein outside the cell generates that disappears. The difference of the viral vectors and other vaccines like AstraZeneca, for example, is that it uses two different reactors, two different vectors. And with the first viral vector, it's going to generate S protein, neutralizing antibodies in this S protein. And also we should not lose sight that this viral vector adenovirus is like an antigen for ourselves. And that generates and it brings on that the antibodies appear. So when this happens on the second one, the antibodies that were developed are not going to recognize this viral vector. So the efficiency of the second viral vector to go into the cells is much more than if it was a second dose of adenovirus 25, which would be blocked. The results have been published. Phase 3 results were published in January. And what was seen in, as we can see in Lancet, is that with this vaccine and this immunization plan with 2 doses separated by 21 days, this brings on high titration of IgG antibodies that are specific of RBD or S protein. It also generates high titles, high titration for different ages, different gender, versus the group that gets placebo. It also generates T-dependent unit with interferon gamma and O4 and 0.8. And if you see the accommodation curve in the cases, we can easily see how the curve of individuals is here versus the ones that get the vaccine with this. This means that the vaccine generates the effector mechanisms that we expect, and it showed efficacy over 90%. So at the end of December, they started to vaccinate first the population in Argentina through the Ministry of Argentina in the province of Buenos Aires, with all the history throughout the year, collaborating, working at the ministry, the municipality, in developing the methods. And they called us to be part of a consortia along with other important labs in our country, like the Foundation Institute Alvar with Dr. Amarwi, and also the Invir's Institute of the School of Medicine in Buenos Aires, in a way to study or monitor the immune response of the population vaccinated in the province of Buenos Aires. First, we saw individuals that got the Sputnik vaccine in the 300 and 100, and currently in individuals that have been vaccinated with Oxford AstraZeneca. 7 public hospitals in the province are part of that, and this samples before the vaccination, 21 days after the first dose, 21 after the second dose, and then 3 and 6. So that is studies that are going to last for a year. And we can see the measure of antibodies, IgG, anti-protein S, and RBD. And titrations are done. And after month 3, we start to measure cellular immunity, and also with interferon gamma and all these cells. 3 to 12 months. And, over 1,200 samples. what I wanted to tell you is that we have results that we have decided to publish in the last revision stage. This cohort of 288 individuals vaccinated with Sputnik, these are all healthcare personnel in our area. And they were broken in 17 to 29 to 59 and over 60. We measure antibodies, anti-S, neutralizing after the first one and after the second dose of Sputnik. And we are at the cutoff point of 3 months measuring cellular immunity. As we start looking at the results quickly, we find something that we have been surprised about. We have seen more work. And what allowed us to divide or break down the vaccinated individuals. Group 1, I'm going to call group 1. They are vaccinated that we call naive. And so sample 0 do not have antibody titles. And when we went to check the patients to see how they were doing, these were individuals that had never had COVID, that were not registered, as I mentioned before. The rest of the vaccinated pre-immunized that showed positive antibody levels before the vaccine, and these individuals were all COVID positive. So this is a graph of the results of the titration of anti-spike titles, which is a baseline condition before the vaccine with baselines or undetectable levels of antibodies, while group 2, they present antibodies in varied quantities, as we have seen in different phase 1, phase 2 papers for the different vaccines. A lot of individuals have pre-existing antibody levels that could be due to natural infections for other type of virus. But when they get the first dose, group 1, naive individuals, we can see a heterogeneous distribution of antibodies. And we can see in different papers, while the vaccinated ones in group 2 that get their first dose, they show antibodies levels that are over and higher than those individuals that would not have this type of antibodies. Here we can see an increase in the titration and the baseline levels after the first dose of about 20 times. So that is that those individuals that were recovering from COVID developed these antibodies during the infection, and what they're doing with the first dose is to intensify the production of the antibodies, and we can see that they develop very high titration of antibodies. When they get the second dose, those that were vaccinated in the first group that had not had COVID before, we can see that 100% they increased the titration of antibodies, but a lot less than patients in group 2. While this in group 2 that get the second dose, the titles does not include and the values continue to be elevated. After the first dose, what we can see is 94% of seroconversion of these patients. If we break them down in over 50 and under 50, we can see that in most of the cases of those that get the first dose, they make the seroconversion for under 60 and a high percentage for over 60. And what's interesting here is that if you compare the titration of the antibodies in group 1, that those are the ones that did not suffer from COVID and they get two doses, this group is less than group 2. Those are recovering that get one single dose, and they have titration of antibodies that are a lot higher than those patients that got both doses. When we analyze neutralizing antibodies, we can see that they were done in both methods. In one of the methods, we use a pseudovirus, vesicular stomatitis, and we can see SARS-CoV-2 virus And we can see pretty much the same thing. The ones that did not have COVID previously, they're not going to have neutralizing antibodies. And when the first dose is received, they develop a very heterogeneous levels of antibodies or titration. And when they get the second dose, that increases the title of antibodies, the same in this other method. And if you compare with group 2, then you have in the baseline condition neutralizing antibodies in different amounts. And this is something that can be seen in many individuals that after COVID, and some have antibodies, some others don't, but it's very heterogeneous. So post-infection response is different depending on each individual. But what's interesting is when they get the first dose of the vaccine, you can see how this increases over 5-fold the type of antibodies while they get the second dose. And this type of antibodies is constant with the same both methods. And there's no significant difference in the variation of the title of antibodies receiving one dose or two doses to ones that have COVID before. Well, we were surprised with this, but then other papers appeared and they are demonstrating, they're showing the same with vaccines in the RBA platform. This is work that compares individuals that get, there were 100 individuals, 88 get Pfizer and the rest Moderna, but you can see the same thing. Some of them have baseline levels of antibodies. When they get the first dose, in 4 or 5 days, these levels increase immediately or very quickly. And individuals that have had COVID and the ones that did not have COVID, it takes a little bit longer to develop antibodies, but after the first dose, the levels are maintained. So those who suffer COVID, not only do they have more antibodies, but they do it faster after the first dose. In this paper, you can see, in this graph, you can see that those vaccinated who suffer from COVID, you can see more intensity that are not severe in the adverse reactions, only systemic, not local. This is also something that was published for Pfizer vaccine, and it shows exactly the same with Sputnik individuals that were infected before. This is over 1,000 individuals that show 35 with COVID. And we see the same. We have baseline levels before vaccination. And then after that, it increases the titration, but not between the first and the second dose. That is, the results that we got with Sputnik in our population are very similar than the ones that we have with the other vaccines in the other population. So these are the first results that are going to be published of Sputnik. So as a conclusion, a dose of Sputnik produces a high seroconversion in a naive individual, over 90%. It increases the titration very clearly of those individuals that have prior immunity or that they are recovering, that have suffered from the disease. The type of anti-S and anti-neutralizing antibodies and those that are recovering is higher than in naive individuals that got both doses. And we did not see an increase in anti-S antibodies in those that are recovering with the second dose. So if they have had COVID, a question that has helped us, and we can see that in other countries, they are deciding likewise, someone that suffered from COVID, is it necessary to give him or her the second dose? Well, the second dose doesn't change the type of antibodies or neutralizing antibodies. So we know how this is done because in other countries, they got one or one single dose of the vaccine. This is important. It has allowed us to adopt health policies at the level of the ministry for those who have suffered from COVID and get the first dose. They have neutralizing titration to donate plasma. This is something that we have seen in those that have received the first dose after COVID. Obviously, we analyze them, but they can donate plasma for treatment of the diseases. So what this shows is that natural infection, it's a boost in the first dose. It's a prime. So what we're doing here in a single dose is to activate the memory and to generate or intensify the titration of neutralizing antibodies, and we know that the virus is highly sensitive to this. So this is all that I have. Thank you so much, and I can answer your questions if you have any. Thank you, Guille. Excellent, your presentation as always. We learned so much. The questions are going to be at the end. I'm going to present now. I'm going to share my screen. You know me. I'm Dr. Liliana Berzotnik. I want to give you another point of view on the response to vaccines, the adverse events. You know that vaccination, together with clean water in the world, penicillin as well, has changed the context of public health. So it was a big revolution in history from this important events for health. It changed. It really changed the rhythm of the infections. So we're not exempt with the current pandemic of new challenges. But when we are facing these new ways of being able to intervene that are of high benefit, we can also be running some risks, which is the so-called adverse effects that are related to the vaccine. My idea, since it's so important to understand what happens as we face these new ways of immunization, because not only do we know what Guillermo has shown, the importance that our hope is in being able to get the vaccine, because this is going to create immunity. And also, we're going to hear about the new variants, the importance of these vaccines being safe. This virus has caused over 120 million people infected in over 200 countries. As I've said, this is a huge challenge. So it is very important to start having the herd effect, because this virus that is in RNA has the capacity of trying to go and dodge our immune system and try to produce new variants. And it's important to have vaccines that have efficacy to try to reduce the infection. There's so many platforms today of vectors or vaccines without vectors, like Sputnik from the Institute Gamaleya, AstraZeneca, CanSino, that we know that these vaccines are giving us like the one in Janssen, a huge challenge, and it's important to have them as vaccines that are based on messenger RNA, like the Pfizer Institute, Moderna, Infectious Diseases Institute, biotech, and inactive, like the ones from Sinovac, and nano, like recombinant of Novavax. In Argentina, I'm sorry, I'm trying to share.
No sé, voy a dejar de compartir y voy a volver a compartir, perdón. A ver, si me deja. Voy a necesitar un poco de ayuda.
I may need a little bit of help, I'm sorry.
Bueno, volvemos acá, que la importancia de
The importance of being able to guarantee a good vaccination is to show the importance of these vaccines that are reliable and that will produce the smallest amount of adverse effects. I wanted to show what happened with the vaccines in Argentina because, as Guillermo mentioned, Sputnik vaccine V has less publications, and they are pioneers in using it in Asian countries and Western Europe. We have a system that has health information that is called, well, it's part of 3 groups. The system, integrated system of health information, where we can report the adverse effects that are produced in front of certain infections and vaccines. All of this is gathered under a commission that will report events that are attributed to the vaccination, and they're going to report it to the National Commission of Vaccine Safety. And today it is an honor for me to be part of this. So it is important that what we do at USABI is to have been able to understand if the adverse effect is related with the product in the develop, with a defect in quality because of an event that is a coincidence that we will discuss, or that maybe can be related with maybe an emotional moment as we see the vaccine, or some moments or something that matches that could be maybe a disease that this individual had. So when we talk about product-related reaction that is with a component of the vaccine, with defects in the quality that is not per se a component of the vaccine, but maybe, for example, something that is related to the production of to the vials that have the different vaccines, errors in vaccination that can be caused by a problem in the manipulation of the vaccine, or an error in the scheduling, like in Sputnik it has happened, to do the second dose instead of the first one, or actions because anxiety before the vaccination, or coincident events that are not related to the disease or the vaccine, but diseases that are concomitant at that time, or maybe even an accident. The truth that we have presented, Savi, we know that 6,500 notification, 24 jurisdictions in our country, that were 49,301 events notified in 6 months, out of which 641 were serious. We talk about serious events when these events cause a death or are life-threatening. They cause hospitalization or prolong the hospital stay if somebody has to be hospitalized. They cause a significant disability, and also in pregnant women, they have a congenital problem. The reality is that the notices that or notifications that we have received, it was a mean age 32.9 to 50 years of age. The age was 40. Most of them were women with a delay in providing notice of 44 days. Most of the notices were in the capital city of Argentina and La Plata, the capital of the province of Buenos Aires. 1.3 of the notices were severe events. The truth is that even though we're very happy, but we would like to have more doses, yesterday we're lucky because we were able to vaccinate 18,870,000 people, out of which 7.93 have 2 doses. The truth is that I have put together this presentation with a report, the 11th surveillance report of CONACYBA group. It was as presented on May 31st, which was just a few days ago. And we presented what has happened in the past 6 months, and you can be sure by June 23rd, I think we still have what I'm going to be presenting. So hopefully we're going to be sharing with you very soon about the new events. The reality is from the cutoff on April 9th on the events of 5,493,153 doses. We can say that most of the vaccinated had Sputnik, then Covishield, and AstraZeneca. But Sinopharm also gave us the largest number of vaccines. Effects were mild to moderate in 90% of the cases. In 280 events, 0.95 needed hospitalizations or were reported as severe. Sputnik vaccine is going to take more room, more space, because it was the first vaccine that we had available for immunization on December 29, 2020. And we were able to see that we had 25,426 events related to the vaccine, which is 744 in 100,000. And most of this were systemic events, but not serious, like fever, headache, myalgias, arthralgia, local reactions, diarrhea, vomiting, and nausea. And other events that is worth to mention, but that most of the events, like with the rest of the vaccines, it would last 48 hours and the patients recover very well. There are very few cases that didn't seem important to show this, because the first publication on Lancet about Sputnik, about some adverse events, 3 patients presented in this report, out of which 1 single patient was related to the vaccine and the other ones just matched them. 1 was indeterminate and the other one was at the same time, because it was the beginning of a scleroderma, the first manifestation of that patient. In Argentina, at the Italian hospital, there was a report on 683 healthcare workers, and 71.3% presented adverse events that were not important. They were mild to moderate. And I'm going to show you that the following chart, 0.1% needed hospitalization. This is a chart. The effects are the ones that I told you. However, we did have 6 patients with anaphylaxis. And the important thing was the report that is not part of our publications. So we are following it very carefully of Guillain-Barré syndrome that has a very important representation in thrombocytopenia. The rest of the events were programmatic errors, lipopenia, anxiety for the vaccination, some other events that just were coincidence. Many patients were suffering from COVID and got the vaccine.
Eventos secundarios fueron más que nada relacionados por la infección
And the
aguda.
side events was because of the acute reaction to the vaccination. And 2 patients that presented pericarditis, 1 and an abortion or miscarriage in the first trimester. And 633 cases being analyzed. What is our experience on AstraZeneca and Covishield? We know of its efficacy. We have several publications. When we know that it prevents hospitalization, admission, ICU, and death by COVID. So that's why it's so important to remove the myths on the adverse effects in these vaccines, particularly in our countries. I know what's happening. I don't know exactly what's happening in the United States. I would like to listen to what's going on in Australia and Europe, because we have this ghost of thrombocytopenia, autoimmune thrombocytopenia. The truth is that with both vaccines, Covishield and AstraZeneca is the same one. We had about 700,000 patients vaccinated, and we don't have a record of adverse events, but the same ones that we have been able to see: fever, with the headaches, with or without myalgia, arthralgia, fever was only one single symptom. And we've had several patients that were suffering from COVID at the time of immunization. Same thing happens with AstraZeneca. In Argentina, this is being manufactured. And in the container of these vaccines, there was a problem. And I believe that this is going to be replaced. So we're vaccinated in an important manner, those patients in Argentina. But what have we seen based on these infrequent events? The thrombocytopenia induced by heparin. The first group by Dr. Graham chart that shows after the administration of 200 million doses about this, and this is produced by Janssen vaccine. This event that was reported after the administration of 2,800,000 doses. What is the mechanism that this is produced? Well, they know that some of the patients have a subjective chart of disseminated intravascular vaccination. Some have plaquetopedia, identified as an immune mechanism that induces a reduction of the platelets that are heparin induced by the alteration of factor 4. And there's no associated risk factors to this event. So The European Agency of Medications, EMA, as the regulatory agency for the medications, and with other allies in the UK, have said that it's a very rare event, and the quality, as you can see, they believe that it's a vaccine that we need to continue giving. The truth of the matter is that I think we need to underscore thrombosis by SARS-CoV-2 is very frequent in natural infections. We had reports of 7.8% pulmonary embolism, 11.2% of deep venous thrombosis, 23% of venous thromboembolism, 1.6% of strokes, and 30% thrombocytopenia by COVID-2, by the natural infection. But also, thrombosis is not infrequent in the general population. In the European Union, there's an annual report of people that develop thrombosis. They have developed thrombosis in the UK. Women with the use of contraceptives and thrombosis with thrombocytopenia caused by the vaccine has 0.004%. And those are the numbers that should convince us to help us physicians, because there's many physicians that are convinced. And it's important to show the infrequency of these events. So both the group of the GAVs, that is the Global Advisory Group in Vaccines for the WHO, they state that under the assessment of heparin-induced autoimmune thrombocytopenia, they're rare. So our ministry continued to say that we should vaccinate, and we continue vaccinated. And this is current data that has not been published. We're about to publish, where it shows that 1,456,740 doses with AstraZeneca are not identified so far, events according to the characteristics of the group of Vito collaboration. So we have not seen autoimmune thrombocytopenia that is induced. Therefore, in this report, we underscore the safety of the vaccine, particularly AstraZeneca, in our recommendations. The vaccine can be given in patients with history of thrombosis. Yes, people with thrombosis history have more risk of having another thrombosis, but not with the vaccine. They do have a high risk with the natural infection of SARS-CoV-2. We can see thrombosis with thrombophilia, yes. Patients with a history of thrombosis or other conditions known as Leiden factor 5 or prothrombotic situations that produce thrombophilia or thrombophilia itself get the benefit of getting a vaccine with the risk of having thrombosis or natural infection. Therefore, it is very important to say that there's no risk. And also, those that have been vaccinated before can get the vaccine. We know that the vaccines are intramuscular. They're very safe, so it's not a contraindication to have the vaccine in anticoagulated patients. Now we go to the other vaccines in my country. Sinopharma in 1.295 million, with a risk of 31.6% of producing fever with headache with myalgia, anaphylaxis in one case. And I think this is important. And GI symptoms like a metallic taste in the mouth, programmatic errors in persons that got the second dose of different vaccine or who had received something else during a different moment, and these were the infections like we had with 26 patients that were suffering from SARS-CoV-2. They were immunized, and the severe effects of hospitalization were not because of the vaccine. And we have 31 cases that are in analysis and data of those patients that do not have the information to be analyzed. I want to show you thrombotic effects related to the vaccine. Throughout the world, we have seen, and with Janssen vaccine, 15 notifications in Spain, Canada, the UK, and where we have seen more and more these thrombotic effects. In France, we have a high number. This is still a low rate against the benefit of the vaccine. This is a safety report for the vaccines in front of the severe events or non-severe in the United States. The Pfizer vaccine has shown on the notice 9% of severe effects. Moderna, a notification on 200 per 100,000, 3% of severe cases, 97 not so severe. In Spain, most were non-severe, same with Pfizer, Moderna, AstraZeneca, that is the most severe. Canada, with the 3 vaccines, they reported several severe effects in patients, 14.9%. Also with France of these events, UK, most of them, the reports were non-severe, but I think most of the effects for thrombosis induced by factor 4 and autoimmune being reported in France. They have reported 22% with Moderna, 16% with Pfizer, and with Oxford AstraZeneca, 27%. So this is something that I think is important to show adverse effects in safety in my country. We know that for those that have received the vaccine, if they have symptoms, they have to be clear that this can be because of the vaccine. But if these symptoms persist for over 48 hours, they should be reported. As physicians, we must know that we must report. And in my country, it's CISA, and CISA, we have to report as SAVIs. It has to be reported with the clinical history, with all the very detailed information of what the patient has gone through, because it's important. Not only is the health of the patient, but the safety with the vaccine and our experience, so we know what to do, whether we continue prescribing that type of vaccine. Right now, we're lucky enough to say that adverse effects are minor. We know that we are facing critical times because this first co-infection that turned into a pandemic takes us to recap and think what is going to happen, because we've had other infections that were epidemics in Asia. We have suffered this pandemic, but very quickly we have to control, like swine flu and the seasonal flu, and that we know that 290,000, 650,000 deaths are caused every year, are caused by respiratory infections. So there's a challenge of the new variants of COVID that increase transmission, they're more viral, and we're thinking what is going to happen with the efficacy of the vaccines. So some produce different things in the health, and we're lucky to have vaccines that are safe. It's still our hope. And I believe that we can continue using it in my country, and I believe that it has been demonstrated to be safe vaccines in the rest of the world. So thank you so much for your time.
Hola, buenas noches.
Good
Hello.
evening.
I am Antonio Condino, member of LAZIT. I would like to thank Liliana and Ekaterina for this kind invitation to present on some data about the vaccination against COVID in Brazil. So I would like to share my screen. So I will be brief because many points of this talk were already covered. So a first concept is about herd immunity. And this we are going to reach, to accomplish in about over 75% of people immunized. This is critical, especially for our primary immunodeficient patients, because depending on the immunological defect, we are sure that they will be unable to produce antibodies. We are not sure about cellular mediated immunity. And eventually, there are those kind of patients that will have a partial response, antibody response, and some of them will respond or not. So vaccinating the general population will eventually provide safety for them. It's a group protection. The coronavirus, as we already have seen, is the principal component that the vaccines are directed against currently is they are directed to stimulate antibody production against the S protein. So here in Brazil, we are using the Pfizer vaccine, which is a genetic vaccine with over 97% efficacy. We are using the Oxford AstraZeneca vaccine, which is a vector vaccine, and will receive very soon the Johnson vaccine. So they are produced over adenovirus platform, that will receive the sequences of the S protein, and they are inactivated. That said, safe for our patients. And we have the Sinovac, the Coronavac vaccine, which is based on the whole inactivated virus. This vaccine has around 50% efficacy, so that means that it reduces the chance in 50% of the person to get infected. However, this vaccine has been proven to protect in a very good way, very efficient way, the severe forms of the disease. And here in our country, we are already detecting this kind of positive effect. So these are today numbers in Brazil. I pulled this from Folha de São Paulo newspaper. So what we have 41% of our population already immunized with the first dose, about 15 receiving the second dose, and these are the total numbers. We have 18 million people here, and unfortunately, 500,000 people dead in our country. About some statistics. So these are the numbers. We are now second place in the world after the United States, and this is the statistics, the relative statistics, the mortality per 100,000 inhabitants. We have an intermediate number. So what happens here is that our country is very populous and very big. I would like to share another screen with you to show some actual numbers here. So this is the number, 68, almost 67 million people already received the first dose of the vaccines. That represents 42.5% of our population, and about 25 million people, which represents 15% of our population already received the second dose. Our state, São Paulo, is more or less on the same proportion. Mato Grosso state is the leader in our country for performing the immunization. We have to consider that this state is less populated compared to the other Brazilian states. This is the total number statistics. São Paulo is the most populous state in Brazil, so has the population similar number to Argentina, for example, and we already gave the first dose to more or less 50% of the people. So what I want to show you here, in one side, we are going relatively, I wouldn't say that much slowly, but we are delayed in our vaccination program compared to other countries that started earlier than us. And the reason is the behavior of the federal government that decided to adopt chloroquine and ivermectin as an early treatment, and did not buy the vaccines in the last year and decided just to do it now in the last time. So let's have a comparison with the world. So this is the world map. In terms of number, this is our statistics. So our index is about 430 doses per 1,000 inhabitants. And here is our population. So Brazil is a continental country and very populous. And it is, I would say, it's really a challenge to perform all that vaccine. So the effort, I would say, to compare is more or less like the United States. However, the United States has a more powerful economy and started much earlier. So although the former president of the United States behaved on the similar way of our current president, anyway, the institutions worked on a positive way, and the United States is performing a good job in terms of vaccination. Afterwards, I would like to listen the comments of our colleague Kate Sullivan, because she lives there and she will have more details. But I would say that for now, we are accelerating the vaccination rate. Just last week, Brazil was the #1 in the world to give the number of doses on a single day. So we are doing the best as we can to overcome this situation. And we have to keep confident that we will control. Actually, I would say that we are currently facing a threat of the third wave to affect us, but apparently is not that bad compared to the first and second wave. So apparently the vaccines already started to protect us, to work on a positive way. And additionally, the death profile is now more focused on people of middle age. So the old people in Brazil stopped to die, and this already reflects the positive effect of the vaccines. So there is hope in this way. If I would be the Brazilian Minister of Health, so the first thing I would do is to stop the bullshit speech as our president currently makes, because his speech is much uglier than what the real numbers and the real efforts reflect the country. So we are actually much better than our president. Unfortunately, Fiocruz and Butantan are working very well and as much as they can to help us control these pandemics. So as I said, I would like to be brief, because most of the explanations about the vaccines were already presented by Guillermo and Lidiana, and I will stop here, and afterwards I will be very happy to answer questions. Thank you.
Alfonso, el video de, please, the record of Dr. Stewart.
Okay, good morning or good afternoon, good evening. Welcome to the LACID COVID seminar webinar. Thanks for the opportunity for inviting me to be involved with this. I'm coming to you from Sydney this morning, and I hope you're all doing well. Thanks to Liliana and everyone in LACID for inviting me to be part of this special event. So as you all know, we are in the ongoing grip of a global pandemic. This is all about SARS-CoV-2 and COVID-19. As of a few days ago, according to the WHO, there'd been nearly 180 million cases of COVID-19 across the world, and tragically approaching 4 million deaths, which is just a dreadful statistic. Optimistically, though, the numbers of vaccines doses that are being rolled out is increasing literally by the day. So not nowhere near what we need, but at least that is certainly moving in the right direction. So this just shows how SARS-CoV-2 COVID-19 has really ravaged the world over the last 18 months. I don't think any of us have lived through anything like this ever. And this just shows, this is starting in January 2020, and this is up to present day. And you see the number of cases that have come through various countries, various continents, various communities, and so on. And it really shows, it doesn't matter where you are, COVID-19, either the wild type strain or now being grouped with all the variants of concern, particularly the Delta strain, is really causing havoc everywhere. So you can see countries like the US have just had this wave that's gone on and up and finally coming down now. France, likewise, up and down, several waves and the like. So no country has been immune, sorry about that pun, immune from the effects of COVID-19 and SARS-CoV-2. So how deadly is SARS-CoV-2 infection? Well, this shows the case fatality rate for many, many different countries. And this is actually the absolute number of deaths in those countries. So something like the US, which has had 500,000 deaths or thereabouts, versus Australia, where I'm coming from, has had just under 1,000 deaths. But you can see the case fatality rate, it ranges, it's between that sort of 1 and 5%. Globally, it's about 2.2%. So about 1 in 40, 1 in 50 people who get SARS-CoV-2 infection are tragically dying. But it sits in that 1 to 5% band, generally speaking. I've listed some of the Latin American countries here for comparison. Australia, India, the US, and so on. A few outliers. Mexico is quite high. Yemen is very, very high. But generally, it's tracking very similarly everywhere. So we knew from the very beginning of the pandemic that there was a number of risk factors for SARS-CoV-2 infection and mortality. The first one that became very obvious was age. So even though the proportions of people who were getting infected didn't really differ that much across each decade, except for very young children, the death rate really shifted according to age. So with every decade of age, the incidence of death increased. So I actually turned 50 last year. So I transitioned from this risk group to this one. So I'm now 3 times more likely to die of SARS-CoV-2 than I was 12 months ago. But you can see this stepwise increase every decade of life, becoming much more vulnerable to SARS-CoV-2 infection. And likewise, pre-existing conditions, health conditions, heart disease, lung disease, diabetes, cancer, and so on, were all risk factors, comorbidities for SARS-CoV-2 infection and COVID-19. But what about people with inborn errors of immunity? You'd think that people who intrinsically have a defect in their immune system due to monogenic variants that compromise a particular pathway or the generation of a particular cell type, excuse me, may be at increased risk of SARS-CoV-2 infection or disease in the context of their pre-existing inborn error of immunity, whether it be immunodeficiency or immune dysregulation. So that was a question that I was involved in, a large team of people last year asking, and this was really built on a couple of case reports that were appearing in the literature very early on in the pandemic, reporting variable disease courses in people who had different types of immunodeficiencies. So the first couple of papers came from Ana Rosa Sorencia and Isabella Quinty, who were looking at XLA patients, B-cell deficient patients. And surprisingly, they found that those individuals were actually fine, relatively speaking. They had very mild disease, and as these titles suggest, recovered quite okay. Then there's a couple of more cases that came out with BTK mutations, XLA, B-cell deficiency, and overall they were relatively mild. Then there was a few more specific cases. So there was a report of very recent of a patient with the STAT1 gain of function, a patient with X-linked SCID, patient with interferon gamma receptor, patient with ARP-C1b mutations, and then NF-kappa B2 haploinsufficiency. So there's a number of case reports that were emerging in literature over the last 18 months or so. So at the same time, or inspired by these as well, myself and many other people, particularly Isabel Metz, Klaus Fornatz, Kate Sullivan, Nazar Maloui, and so on, started thinking about this at a global level, trying to get a snapshot of what SARS-CoV-2 is doing in the context of people with inborn errors of immunity. So we ran, this was again done with ESID and the iOS committee and many other societies. LACET, of course, was part of this. Sent out a survey to clinicians trying to gauge what's the impact of SARS-CoV-2 on patients with inborn errors. So we ended up getting clinical information from 94 patients. They were very much from across the world, none from Australia, fortunately, but many from Latin America, from Europe, from North America, from the UK. The death mortality was about 10%, and I'll go through this in a little bit more detail in a moment. So that was our first glimpse of what SARS-CoV-2 is doing to the PID community. And since then, or along with that paper, several other sort of cohort studies were published coming from specific countries. So, for example, this paper is out of Iran, this study is from Mexico, this study is from the UK, this study is from Israel. And we've also had much more focused studies, cohort studies, a couple of studies straight out of New York, for example, looking at patients with primary immunodeficiency or CVID. And interestingly, the mortality rate from these different studies varies dramatically. So we reported about 10% mortality. That was confirmed by some other studies, but other studies found it was much higher. So, for example, this study out of Mexico was about double that, 20%. Likewise in the UK. Interestingly, the study in Israel, they had no mortality at all. So 20 patients, no deaths. But then Iran, very high. So it sort of depended on the location, the study, the types of diseases they had. But collectively, we're starting getting a picture of what SARS-CoV-2 infection was doing for patients with inborn errors. So from these 7 or 8 studies, just looking at the numbers, there's about 260 patients with a vast array of inborn errors, and the mortality rate is about 13%. Now, of course, that's much higher than what I showed you before for the global level. But just bear in mind, when we did this last year, the mortality rate was actually much higher, particularly in parts of Europe, in the US, where it was in that 5 to 10%. But overall, 13% mortality rate based on the numbers that keep appearing in the literature reporting in patients with inborn errors and SARS-CoV-2 infection. So I'm just going to talk a little bit about the study that I was involved in to give you a bit more specific information about that study. And I realize this is probably a bit of an unconventional way to show the data, but I actually like the way this is presented. This is also, I should acknowledge, Georgia Bicoli, who worked with Isabel Metz, who did a lot of the clinical analysis and so on. So this is a breakdown of our cohort. Most patients, over half of them, had an antibody deficiency. And then there were a number of patients who had combined immunodeficiencies, phagocytic defects, immune dysregulation, and so on. But the vast majority were antibody defects, so CVID, XLA, other types of antibody deficiencies. So this is broken up into the type of inborn error by color, and then by age around the wheel here. So we've got youngsters under 18, 18 to 55, and then over 55. And then each age group is broken down as to whether they had or did not have comorbidities, and then whether they needed to be admitted to hospital or not. And if they were admitted to hospital, if they were on ICU, needed ICU or needed some type of ventilation. So the first thing you can see is many of the patients who succumbed to SARS-CoV-2 infection had antibody deficiencies. Many of them were in intensive care. So as shown here, for example, many of them were in the older age group, over 55. So 4 out of the 9 deaths that we noticed were in this over 55 age group, and most of them had comorbidity. So it was a little bit like what we saw in the general population, the older patients who had pre-existing health conditions. Interestingly, there were no deaths in patients with combined immunodeficiencies. So these are conditions like ZAP70 deficiency and some other monogenic defects that affect T cell function. And again, over here, when you look at some of these other types of inborn errors, the patients who did succumb were in ICU and often had comorbidities. So whilst the numbers were slightly higher in terms of mortality, the predisposing factors were pretty similar to the general population. So that was an interesting snapshot of what was going on. I must say the ages, because we were identifying younger patients than in the general population, the numbers of patients that needed ICU admission and the age range was much lower, higher for ICU admission, lower for age than the general population. But what was really interesting is sort of the information we could glean from this cohort. Now, whilst 94 is a pretty good number, as I said, there was lots of different types of gene defects or causes of disease amongst those numbers, but we could make some very broad generalizations. About 30% of patients were asymptomatic. They were picked up to be SARS-CoV-2 positive for incidental reasons, like they were traveling or they were needed to go to a hospital for surgery or something like this, unrelated to their inborn error. They were tested and found to be SARS-CoV-2 positive, but actually asymptomatic. So about 30% of patients were asymptomatic or mild disease. And many patients actually had, some of the patients that had mild disease were found to have autoinflammatory conditions. So Aicardi-Goutieres syndrome, for example, FMA, familial Mediterranean fever was another one. Patients who had XLA or B cell deficiency had mild disease. Some of the combined immunodeficiencies, as I said, CGD, and patients who had impaired IL-6 signaling, so autosomal dominant hyper IgE syndrome due to STAT3 mutations, autosomal recessive hyper IgE due to PGM3 mutations, and STAT1 gain of function. They all had mild disease. The more severe diseases included impaired NF-kappa B signaling, interferon gamma receptor, and as I said here, some of the older CVRD patients who had those comorbidities. So thinking of that data gave us an opportunity to think more about the consequences of SARS-CoV-2 infection, what immune determinants might be driving some of the pathology, and how some of the therapies that are being used could be interpreted. So the first one that struck us was the IL-6 signaling. So as I said, we had a couple of patients. One had STAT3 loss of function. We also had another patient with STAT1 gain of function, which has a lot of clinical and cellular features like STAT3 dominant negative hyper IgE syndrome patients. And we also had a patient with PGM3 deficiency, this group of patients who have been shown to have impaired signaling through the IL-6 receptor. So arguably, that may suggest that STAT3 signaling, probably IL-6 signaling, drives that cytokine storm that we're so familiar with, and hence the desire to test tocilizumab as a therapy for SARS-CoV-2. But this probably provides some explanation as the mechanism of immune pathology in the context of IL-6, STAT3 signaling driving some of that cytokine storm. And if you can't signal through IL-6 for mutations that affect that pathway, or because the variant, in this case PGM3, prevents expression of GP130, you might be a little bit protected from disease. Again, I touched on this before, BTK mutations, XLA. Even before we did this work, there were studies that were being published on the use of BTK inhibitors like ibrutinib and others to treat SARS-CoV-2 in the general population. There were studies that were looking at SARS-CoV-2 in patients who were on BTK inhibitors because they had B-cell malignancies, or there were trials that were ran to look at the effects of BTK inhibition. And this did show some promise. And I think the interpretation of this is, excuse me, it's not because BTK inhibitors are targeting B cells, it's probably targeting monocytes, myeloid cells, which are making that IL-6. And it has been shown that BTK is important downstream of TLR, ligand interactions to drive cytokine production by myeloid cells. And that's sort of shown here. What this is looking at is the level of phospho-BTK in healthy donors, and then in SARS-CoV-2 infected individuals, showing that BTK is activated in their monocytes, but not in their B cells. It shows again that the monocytes that are either resting or stimulated, their production of IL-6 is increased in COVID-19 patients compared to healthy donors. But then this doesn't happen in B cells. So the monocytes do make a lot of IL-6. So BTK inhibition is probably attenuating this somewhat. So there's probably good evidence there for why there's mild disease in some of those conditions. A lot of information on this, but this is just something else that wasn't really a finding from our study, but I'll come to the significance of this in a moment. Other aspects of SARS-CoV-2 in the general population, these 3 studies, one in PNAS, one in Nature Medicine, one in Science Immunology, have all shown that dramatic activation of the complement pathway is associated with severe COVID-19. So this Science Immunology paper shows this is otherwise healthy individuals, not PID patients, those who are in ICU or needed invasive mechanical ventilation. Their levels of C5a, for example, were much, much higher than COVID individuals who didn't need ICU admission or ventilation. And likewise, these are individuals who are uninfected or then who had low, medium, and high levels of viral infection, looking at all these complement factors. So on the left-hand side here, you can see there's this almost dose-dependent increase in these complement mediators. And over here, these regulators of complement decline with disease severity or level of infection. And the reason I'm showing you this is because there was this paper that looked at hereditary angioedema, which is a disease characterized by impaired complement activation. It was a small number of patients, 13 patients, but there was no deaths. So again, suggesting that whilst you may think a defect in a major pathway, whether it be IL-6, whether it be BTK, whether it be complement, may be a problem, those conditions may actually protect patients from severe disease. So what else can I tell you about inborn errors? So many of you would be familiar, and many of you are probably part of the COVID human genetic effort, which was set up by Jean-Laurent Casanova and Helen Su in the US. At the beginning of the pandemic, they brought together a global team of researchers, clinicians, DNA sequencing hubs, infectious disease specialists, with the mission to understand the genetics of severe COVID-19, or actually, on the other hand, the genetics of resistance to SARS-CoV-2 infection. So really trying to understand what genes influence our immune system and how we as individuals respond to SARS-CoV-2 infection. So their hypothesis is based on previous studies actually done by the Casanova lab and by Helen Su's team at NIH, where they, plus other groups, of course, had identified variants in genes in the type 1 interferon signaling pathway were actually causal or pathogenic in the context of severe infections with some other viruses. So, for example, herpes simplex virus, we know, is really prominent in individuals who have mutations that affect the TLR3 type 1 interferon signaling pathway. Individuals who have really bad problems with live attenuated viruses have compromised signaling through type 1 interferon type 1 interferon STAT2 signaling pathway. Influenza A virus, likewise, is impaired in people with IRF7, IRF9 mutations. And then other infections like rhinovirus and so on can be impaired by MDA5 mutation. So there's this suggestion that there's likely going to be people in the general population who have variants in this pathway who will be susceptible to SARS-CoV-2, but because this was a novel pathogen, never been seen before, we just didn't know who those people were. So to cut a long story short, the COVID human genetic effort published 2 papers in Science last year, about September last year, indeed showing a really important role for type 1 interferon responses in protecting the host against severe COVID-19. So the first study on the left here identified genetic defects in the type 1 interferon signaling pathway. So some of those genes I just mentioned then were found to be mutated in about 3.5% of individuals with severe COVID-19. So mutations in the components that are important for producing type 1 interferons or responding to type 1 interferons. But perhaps what was more striking was this paper by the same teams who found not gene defects affecting the interferon pathway, but actually found that individuals produced autoantibodies that neutralized endogenous type 1 interferons. So instead of having a genetic defect that compromised signaling through type 1 interferons, there was 10% of individuals, interestingly, mostly males, who produced autoantibodies to type 1 interferons, and that actually blocked the protective effect of these cytokines, thereby both studies showing this important role for type 1 interferon signaling in host defense against SARS-CoV-2. So this is interesting because one of the patients that was in our study actually had GATA2 deficiency. And a feature of GATA2 deficiency, of course, is a lack of plasmacytoid dendritic cells or interferon-producing myeloid cells. And we would speculate wildly, but speculate that the cause of severe disease in GATA2 deficiency due to SARS-CoV-2 infection results from a lack of interferon-producing cells and that impaired innate immunity in those individuals. So continuing with the interferon pathway and studies, one of the early papers in this space reported that a patient who had APS1, so APOSED, air mutations, actually had very severe pneumonia. This is a patient from Italy, which was interesting because, as many of you would know, a feature of APOSED, apart from autoimmune disease and candidiasis, is actually the production of high levels of antibodies to cytokines. And one of those cytokines are the type 1 interferon cytokines. So this was followed up very recently by this study, again, that involved Joel Casanova, Helen Su, but also in this case, Mikhail Leonakis at NIH. And they had a cohort of 18, I think, APOSED patients. And this shows that they all have antibodies against all the type 1 interferons, except for kappa and most epsilon. There's a few healthy donors on the side here. So they studied this cohort of patients to see what would happen to them. And they all had COVID-19. And what was striking was that they had, I think they must have had more, they had 22 patients, I think. 19 of the patients were hospitalized, which is a very high hospital admission rate. 68 needed ICU, again, which is very high, and 4 of them died. Now, of course, small numbers, but that's a pretty high death rate. Again, just reinforcing this role for type 1 interferon in protection against SARS-CoV-2. So if you have a mutation in the pathway or autoantibody, you are predisposed to severe COVID-19. And this, of course, has led to a couple of experimental efforts to test the efficacy of interferon therapy as a treatment for SARS-CoV-2. So this is a nice review written by Isabel Metz and colleagues on behalf of the COVID human genetic effort that was just published in Journal of Clinical Immunology. That's a review article. But these two papers here actually did try interferon alpha or interferon beta therapy in a couple of patients with inborn errors. These individuals had mutations in, as it says here, TLR3 and IRF3, and this individual had mutations in NEMO. There was, of course, they're not controlled studies, but there was anecdotal evidence that the outcomes of these individuals treated with interferon was much better than generally people who were not treated. So again, reinforcing the role of interferon. So I'm just going to spend the next couple of slides and finish my talk just talking about what do we know about actual immune responses to SARS-CoV-2 in people with inborn errors of immunity. So this is from a nice review from Alexandra Sete and Shane Crotty from the La Jolla Institute of Allergy and Immunology in California. And this is like textbook cartoons showing that early on your innate immune response responds, and then over time your adaptive immune response kicks in the face of a generic infection. Antibodies come on, CD4, CD8 T cells respond. In SARS-CoV-2 infection in the general population, people who have mild disease and cover, who have this lag time of when viral load is increasing over time, declines eventually, your innate cells do something, and then your adaptive cells, again, start making T cells and responsive B cells and antibodies, and things go okay from there. But though with severe SARS-CoV-2, viral persists, T cell dysfunction seems to be not so, T cell dysfunction seems to be prominent, antibodies are variable, and innate immunity in the context of like a cytokine storm keeps persisting. And this is showing it a little bit more detail here. This is, again, models of what we think the immune response in healthy donors to SARS-CoV-2 looks like. So this is your average COVID response. It's most of the viruses in your upper respiratory tract. Some gets into the lungs. In fatal COVID-19, the virus sort of goes everywhere and is very problematic in your lungs, obviously. The T cell response is predominantly CD4 T cells, but there's a contribution by CD8 T cells. And your antibodies, you have antibodies of different specificities, of course, but the most important antibodies are those against the spike protein, and of course, those that are neutralizing. So there's one study came out very, very recently from Mike Keller, who's in DC, and this actually studied antibody and T cell responses in SARS-CoV-2 patients who had antibody deficiency. So they studied 5 individuals from one family. Several of these individuals, the ones shown in black, actually were SARS-CoV-2. The patients shown here, P5, and a couple of the other ones were found to be SARS-CoV-2 positive, but those in the black circles actually had pre-existing specific antibody deficiency, hypogamma, or CVID. And they also looked at 2 other CVID patients unrelated to this family, and then were able to track their immune response following SARS-CoV-2 infection. So the top here, this shows antibody responses, and here's the antibody deficient individuals. So you can see here P1, P2, P4, and P6 are antibody deficient. P6 and P7 are the CVID patients unrelated. And then these are the immunocompetent individuals, 3 and 5 there from this kindred over here. So basically looking at antibodies against the nucleocapsid or spike, this is what the controls look like. From these 5 individuals here, except for this P6 who was hypogamma, they could detect antibody responses to the nucleocapsid as well as to the spike protein, which were not too dissimilar to levels in the immunocompetent controls. And then this is looking at CD4 T cell responses, again, in responding to different peptides corresponding to the membrane, envelope, nucleocapsid, or spike proteins of SARS-CoV-2. And again, for the immunocompetent individuals shown in the red, compared to the antibody deficient individuals, pretty similar responses, no significant differences. And in fact, there's this trend for higher responses in the antibody deficient individuals here. So at least in the context of natural viral infection, with the exception of this one patient who was A-gamma or hypogammaglobulinemic to start with, at least the short-term response was pretty intact. So as the authors concluded here, this shows robust T cell activity and antibody responses against SARS-CoV-2 in patients with antibody deficiency, generally speaking. But what about vaccination? We're hopefully moving into that phase where we'll see many fewer cases because the vaccine is being rolled. As I said at the beginning, billions of doses have been distributed. So this is a really nice study that just came out as well in Journal of Allergy and Clinical Immunology from David Hagen in Israel. They were lucky enough to study 26 patients with inborn errors, and they looked at these patients 2 weeks following their second dose of the mRNA Pfizer SARS-CoV-2 vaccine. And again, like some of those studies that looked at SARS-CoV-2 in inborn errors patients, this is a mixed array of mutations that cause these conditions. So this is first looking at antibody levels. So there's a lot of data here, but I'll just go through it. These are uninfected healthy donors, so people who have never seen SARS-CoV-2 before, so they don't make an antibody response. These are patients with inborn errors of immunity, and you can see that it's quite variable, and I'll come back to that in a second. These are inborn errors, patients who have been vaccinated, I should say. These are healthy donors who have been vaccinated, and these are people who had SARS-CoV-2 but are now convalescent. So comparing this color and the yellow symbols, you can see there's pretty good responses, slightly higher for the healthy vaccinees. But what's important to note here, and I should say this is for viral inhibition as well. So again, there was a range of responses in the patients, but generally speaking, it was much higher than uninfected individuals in the healthy population. But key point here is that 4 of the patients had XLA, and 1 patient had been treated with rituximab. So they were B-cell deficient, and variably antibodies were not being able to be produced because they had no B cells. So overall, when you take those patients out, 81% of their cohort, 18 out of 22, were found to make antibodies against SARS-CoV-2 vaccine. There's a bit more information here just showing there was good correlation between the levels of IgG produced against SARS-CoV-2 and the neutralizing capacity. And when you break it down into the different types of inborn errors, again, these are the patients who have no B cells, they're B cell negative, so they don't make a response, obviously. These are patients who have like autoimmune diseases, so these were individuals who had ALPS, for example. Complement deficiency response was totally intact. And then there was a bit of a difference between CVID patients depending on age. So those under 50 generally had a better response than those over 50. And this is one patient with an inborn error who was infected, but then recovered. So basically, when you look at it in that context, at least in the short term, the antibody responses were encouraging, to say the least. They also looked at T cell responses, and this is looking at cytokine secreting cells in vitro in response to a whole range of peptides. And again, same layout here. These are before exposure, and then these are the patients. These are healthy vaccinees, and these are convalescent individuals. So again, in response to the vaccine, most of the patients could produce a T cell response. It was actually 73%. And then again, when you look at it, breaking it down, whether these are patients with inborn errors who do or do not have B cells, those who don't have B cells were capable of making a T cell response, which was pretty comparable to the healthy vaccinees and slightly better than recovered healthy donors who were SARS-CoV-2 infected. So basically, the conclusion from that is that, at least in the short term, in other words, the first study that looked at natural infection and this study that looked at vaccination really only looked weeks to months after infection or vaccination. They could detect robust humoral and cellular immune responses in patients with inborn errors. Caveat being, of course, if you don't have B cells, you're not going to make antibody response. The vaccines were safe, so vaccination should be encouraged for inborn errors of immunity in general. But obviously, these are early days, so we need to do much larger cohort studies and longitudinal studies to see what the durability and the efficacy of vaccination in inborn errors against SARS-CoV-2 really is. So I'll stop there, and I just want to acknowledge everyone that was involved in that international study that I was involved with. Again, particularly Isabel, Georgia, Kate Sullivan, Klaus Vornatz, and many others on this list, and the patients and their families. And I'll stop there, and I'll thank you again for your time, and I'll take questions if there's any. Thank you. Okay, good morning.
Hi, Kate.
Hola, buenas noches. My name is Kate Sullivan, and I am going to present in two parts. I have a little piece at the beginning that is 10 to 15 minutes long. Dr. Tanguy presented a little bit of the data, so we'll be able to go through that part a little bit more quickly. And then a colleague of mine is going to present a case that is really meant to be a cautionary tale. We don't have an answer for it, but I think the more people we vaccinate, the more we will see the rare and unusual consequences. As Dr. Bezrodnik talked about, there are adverse events with all vaccines. There's nothing unique about these vaccines. So let me start. I'm from Children's Hospital of Philadelphia. I'm a pediatrician, and my colleague, Dr. LaCava, is trained in internal medicine. And so why are we convening this? You can think about it really simply of the patients that are immune deficient. We worry if they're going to get protection. What will we do with our patients? How can we protect them in the face of this pandemic? And then on the other side of the coin, we want to understand, is there a group of people that should not get the vaccine because the risk of adverse events is very high? So that's really what I'm going to talk about with some data, some modeling, and some conceptual approaches. So this is a study that I really love. Actually, a friend of mine, Stanley Plotkin, put out a very similar study, but I like the graph in this one better. So you can find your favorite vaccine. I know every country has their own set or single vaccines that they're using. Let me explain how this graph is set up. So on the y-axis, this is the protective efficacy reported in clinical studies. Across the bottom, the x-axis is neutralization in vitro. So you take the serum from someone who has been vaccinated and you put it in a test tube with pseudotyped virus and you ask, does it neutralize? So that's sort of the gold standard rather than ELISA to look at antibody production. So what you see is an incredibly close correlation between the clinical efficacy on the y-axis and the in vitro neutralization. So at the upper right, you see the most effective vaccines, which are the mRNA vaccines and Novavax. And then way over on the left, you see the ones that are less effective. Sputnik did not appear in this study, so I can't tell you where it falls. So it says, in essence, that there is a strong correlation. You can almost predict the clinical efficacy based on the in vitro neutralization. Not that we're going to stop doing clinical trials. Of course, we will do clinical trials, but there is such a close correlation there. Clearly, you can get a sense. And the reason I'm showing you this is the foundation for a set of models that were done by this group. So this is from the same paper, and it's asking the question, can you predict efficacy against the variants? Can you predict efficacy against the variants based on the in vitro neutralization? Sorry, my AirPods are running out of juice and I may have to switch over. So sorry about that ahead of time if I have to switch. So again, if you look at this graph, you're looking at efficacy against the wild type vaccine, and that's the dotted line. And then they imagine 3 different types of variants where you have altered binding in vitro. And based on the graph that I showed you before, they're predicting clinical efficacy. So why am I sharing this with you? Well, we all know variants are coming. They're already here, and there will be more. We're not done. It's not like Delta is going to be the last variant we see. And so we need to understand how effective the current vaccines will be against those variants. And so using the modeling that I've talked about, you can see at very high rates of efficacy with the vaccine, you don't really lose much. You don't lose much with the variants. But once you get to lower levels, the vaccines that are not as robust, they are not as strong, then you start to see decrements with the variants because you lose the in vitro neutralization, and that translates to compromised clinical efficacy. Now, the other question that Stu Tangi brought up that is certainly relevant for all of us is, what does this mean over time? So Stu presented data showing that immune deficient patients can produce antibody. The question is, will it last as long? So of course, we can't predict in any one patient, but let's just look at the general population for a minute. So this is based on 3 different decay models. So there's only 3 sets of clinical data about the decay of antibody right now. That will change dramatically over the summer, our summer, your winter. But for right now, what we know is out to 6 months based on a study of natural infection of nurses in the United Kingdom, and then the 2 mRNA vaccine studies where they followed people out to 6 months. So based on those data, again, using this modeling system, you can see that out to 3, 4, even 5 years, you still have good efficacy against severe disease, which is the dotted lines, but you have much less efficacy against mild infection. And that is exactly my experience at my hospital. There have been a number of breakthrough infections, and they have all been mild. We all got vaccinated as part of a hospital program back in January and February, and there have been some breakthrough infections, but all of them have been mild. In fact, some were picked up through contact tracing. So that is my experience, and that is typical for other vaccines as well. As your immunity wanes, you retain protection against severe infection the longest, and you lose protection against mild infection. All right, so how would I summarize this? There's nothing really fancy other than I've shown you some lovely models that kind of make real the issues that we're facing. So more antibody is better, right? If your starting level is higher and you decay at a certain rate, you're going to end up with more in 5 years. So more antibody is better, but some antibody is better than no antibody. And then protection is going to be unpredictable. So everyone wants to know, do we need booster shots? What's going to happen to our patients? We don't exactly know, because we don't know what variant there will be next year. We're on Delta now. What are the future Gamma variants going to look like, and how will that impact the neutralization? So we can't predict the variants. And then all of us involved in immunology know that our patients can have unique trajectories. So there's the population average of the decay rate of the titers, but we know that there's a lot of variation, and our patients tend to lose immunity faster. So it may be that there are subsets of our patients that need boosters before the general population. And just as an example, in specific antibody deficiency, we know that some patients with specific antibody deficiency mount responses right after their vaccine, but they lose it in 6 months. And so that's just one example of how this personal trajectory might play out. Well, is there more than antibodies? So Stu Tangy talked about this a little bit. Let's just look at a tiny bit of data from mice. There are very similar data from other animal models, but I happen to like the way this study was done. So let me just explain it to you. So at the top, this is a mouse that is infected with SARS-CoV-2. The mouse recovers. In fact, mice generally recover from SARS-CoV-2. So this is a mouse that recovers from the infection. They take serum from the mouse and T cells from the mouse and put it into another mouse that doesn't otherwise have any immunity. So now the only immunity in these second mice comes from what they got from the previously infected mice. And then they infect those mice and they ask what happens. So you're trying to dissect how much does antibody all by itself protect, and how much does T cells protect all by themselves without antibody, which is of course impossible to do in people, other than you could look at patients with XLA. So this is the control. So the black lines, the black bars here are the mice that got nothing. So the first graph that you see on the left, they're doing PCR for the virus, and the graph on the right, they're actually recovering virus and growing it. So if you look, the black bar is what happens when there's nothing transferred into the recipient mice. The red bars are what happens when they get T cells. So by PCR, it doesn't look very dramatic, but if you look at the virus recovery in the right-hand graph, remember this is a log scale on the y-axis. It's actually almost 1,000-fold less just transferring T cells. But of course, you get the best immunity by transplanting antibodies from the previously infected mouse. So what does this say? Well, antibody is certainly what it's all about in terms of protection from infection. There's no question that protection from infection is largely mediated by antibody, but for our patients that have XLA and no capacity to produce antibody, they will still make a T cell response to the vaccine, and that will protect them a little bit. Maybe not as much as antibody, but it will offer them some protection. So what does this mean for people with CVID? Well, they can make antibody, and some protection with even a little antibody production is helpful. Stu showed you the data about the patients with common variable that were vaccinated. I'll show you that as well. And certainly even lacking strong antibody production, they will still get T cell production. And so we are recommending that our immune deficient patients get vaccinated, even if they have an antibody problem. So this is the data that Stu shared with you. I do think it's really important. This is the study from David Hagen. And so I'll just remind you of the punchline. I won't go through it in the same level of detail. So the first column is what people look like before they got the vaccine. So the pink symbols are people before vaccination. So no antibodies, makes perfect sense. The dark red symbols are immune deficient patients post-vaccine. So Guillermo showed you what happens after 1 dose, after 2 doses. These are patients after 2 doses. And they compare it with healthy people after the same vaccine regimen and people who are convalescent from natural infection. So who responded? That's what we want to know. So Stu already pointed out there were a few that had no response. They were largely people with antibody problems. But look, most of the patients with common variable did respond. So what is my experience? Completely aligned with that. I have 3 patients with common variable who got the vaccine. They all responded. 2 patients with specific antibody who responded, and I have 1 patient with XLA who did not make antibody. So my personal experience exactly lines up with that paper. So what about risks? So none of the licensed vaccines, or even vaccines that seem close to getting licensed, are replicating virus. So they're not capable of causing infection. Yes, the coating is made from a virus in many cases, but all of them have been crippled. They do not replicate. They cannot cause a viral infection. So really what we're talking about is the classic adverse events that Dr. Bezrodnik talked about. We have to be mindful of that. Our patients will ask us. They are already asking about it. So what about interferonopathies? These vaccines induce really high levels of interferon, and so should we restrict our patients with a Cardi-Gouttiere? So we do. I know not every center does. And similarly, we do not give it to people with severe mitochondrial diseases because infections and fever can tip them into a metabolic crisis. But we certainly give it to the well-controlled older patients. So those are 2 subsets where we do restrict it. That's not very typical for our immunology clinics, but those are 2 unique subsets where we restrict it. But is there more? And so Dr. LaCava is going to present a case. I said a few minutes ago, we don't have an answer as to what happened to this young man, but we do believe it is vaccine related. And it points out what Liliana was saying, where we have to be very mindful about the adverse events, and we have to track them. So the rare events like the one you're going to hear about, those certainly aren't happening all the time, but we need to understand what is happening in those patients so that we can prevent it from happening again. So I'm going to stop sharing, and Dr. LaCava is going to present a case. Anthony, can you share your screen? Excellent.
Can everyone hear me and see me?
Yes, very nice.
Can everyone see me?
No, we just have an A.
I'm trying to start my video, but the host has stopped it.
Oh.
I'm not sure if they want to start my video if possible, but.
Liliana, can you help Anthony get his video started? And Anthony, maybe,
Perfect.
there you are.
Perfect, there we go. Good evening, hola, buenas noches. My name is Anthony LaCava, and I'm a fellow in allergy and immunology at the University of Pennsylvania. And I'd like to thank my colleague, Dr. Kate Sullivan, for inviting me to speak at this lovely talk. And I'd also like to thank Liliana and Ekaterini for arranging this wonderful conference. It's really special to take part of this cross-cultural transmission. And what I wanted to do is to take time to discuss a vaccine event, a patient that Dr. Sullivan and myself had taken care of at the Hospital of the University of Pennsylvania. And really, this is a case that we can all learn from, and is sort of a warning for all of you about possible rare adverse effects that can be seen with this vaccine. So I'll just start with the initial history of present illness. So this was a 20-year-old male who was previously well and received the first dose of the Pfizer mRNA COVID-19 vaccine. And within 2 hours of receiving the vaccine, he developed fatigue, vomiting, diarrhea, and lower abdominal pain. And then within 24 hours, he noticed whole body stiffness and urinary urgency. And at 2 days, he had developed gross blood in the urine, and he developed some tinnitus, some hearing loss, and had a pustular rash that erupted in his face and chest. And this was a patient that had never had a prior issue or any reaction to vaccines in the past. He had not had any recent sick contacts, preceding colds, any diarrheal symptoms, no new medications, no over-the-counter analgesic use or supplements, travel, recreational drug use, or any sexual activity. And actually, both of his parents had tolerated both doses of mRNA COVID vaccines. So upon initial presentation to the hospital, he was found to have a Tmax of 100.3 degrees Fahrenheit, and he was tachycardic in the 120s. He was with a normal blood pressure and with normal oxygenation on room air. And on physical examination, he was found to have an acneiform rash on his face, also on his chest. Though his lungs were clear, he was tachycardic. And though his abdominal exam was normal, he was found to have a pustular rash all over his face, his chest, his upper back. And though he had a normal neurological exam, he did have both ears, he had complete hearing loss. So some of the labs that had been obtained right on his initial presentation were pertinent for neutrophilic leukocytosis. He was lymphopenic. And as soon as he had walked in the door, his creatinine was 3.8 with a prior baseline of normal. So he was already in renal failure. He had a normal lactate, he had a normal INR and ferritin, but he did have some elevated inflammatory markers with an ESR of 29 and a CRP of 2.5. Workup also included HIV, syphilis screen, Epstein-Barr, CMV, hepatitis panel. Those were negative. And then he had a rheumatologic autoimmune screen, which was normal for ANA, ANCA. He had a normal LDH and haptoglobin. He had normal quantitative immunoglobulins and a normal SPEP. And the team did evaluated him extensively for infection, and the workup was negative. He had 3 COVID swabs that were normal. He was swabbed for flu, and that was negative. He had a rapid strep that was negative. His urine and blood cultures showed no growth. He had stool studies that were normal. He even had fungal blood cultures that were negative, and he had a lumbar puncture that was normal, and the CSF cultures showed no growth. Because of his hearing loss, he ended up getting an MRI of his brain, and that showed some punctate areas of ischemia in multiple vascular territories. And because of his kidney injury, he underwent a renal biopsy, which showed glomerular hemorrhage, which is usually immune mediated. And the skin was biopsied, and that showed suggestions of inflammatory folliculitis. His bilateral hearing loss was really of an unclear cause. So in summary, we had this 20-year-old male with no medical issues who acutely had a febrile inflammatory response after a vaccine, manifesting with rash, hearing loss, hematuria, and renal failure, with no pathogen that was identified and no signs of autoimmune disease. He did have an initial cytokine panel that showed an elevated IL-2 receptor, an elevated IL-10, and elevated IL-13, which is indicative of some T cell activation. And he also had an elevated CXCL9 level, which is indicative of gamma interferon effects. He also had a whole exome sequence that showed some variants of unknown significance in the C1s gene, though he did have normal complement studies. So his hospital course, so he had received high-dose steroids, plasmapheresis, IVIG. He had required renal replacement therapy. He also required pressors. He even developed a capillary leak syndrome with bilateral pleural effusions requiring a chest tube, pericardial effusion with tamponade requiring a pericardial window, profound anasarca. And then he also had developed alopecia areata. So what happened to this guy? So hearing loss is associated with therapeutic interferon, and alopecia is also associated with local interferon production and usually is treated with JAK inhibitors. Renal failure has also been reported with interferon treatment. Capillary leak syndrome has been reported with interferon therapy, and lymphopenia can also be caused by interferons. And we keep asking ourselves this question, like, why this guy? An article by Gogas et al. Published in Anti-Cancer Research in 2004 about adverse effects of interferon alpha treatment in oncologic patients had shown a profile of the type of side effects that these patients can develop. And really, our patient had almost all of these. So, we think that the mRNA vaccine had caused an interferon response, which ultimately contributed to his inflammatory state. So in conclusion, we don't really know why, but if we saw this again, we probably would initiate a JAK inhibitor treatment for this gentleman. Thank you.
Any questions for Anthony before he closes his presentation? No questions, Anthony. Would you please stop sharing your presentation?
Yep.
So we can. Thank you very much for all the great presentations. I'll start making the questions. I'll have to translate some of the Spanish written questions, so maybe it's not so good. The first question is to Professor Guilhermo. Armando Cruz questions, how much time after COVID? Please, how much time after COVID?
I just have listening questions.
Okay, first for you, Tanji.
Oh, I've got a question. Yes.
I don't know if you have any more questions. Do you have? You had a question from Saul and you answered. Maybe you can answer alive for us.
Sure. Can you hear me okay?
Yes.
Yes.
Great.
Doctor.
Great. So yes, Sol had a question about NLRP3 gain of function patients who you would think might be susceptible to hyperinflammatory consequences of infection, but they haven't seen severe COVID-19 in any of those cases yet. And there have been some big studies of large cohorts of PRD patients in Mexico recently and so on. So I don't have a clear answer to that, but I was just wondering if the patients are on JAK inhibitors, maybe that's enough to attenuate the severity of the disease, the intrinsic disease of the interferonopathy, but maybe that's also enough to sort of poise their immune system, particularly their innate immune system, to be ready to go and be protective. But I don't really know. But that would be my wild speculation.
Any more questions for Professor Stuart Tanguy? Do you want to say something else for us? Once we don't have any more questions for you?
Stuart or Kate, can the patients with immunodeficiencies with receive the vaccine, they can to suffer the new?
I imagine there will be breakthrough infections, just as there will be in the healthy population. I'm not familiar with any of those cases yet. Kate's probably got a better visibility of this than I, just because there's many few people in Australia and many fewer cases in Australia. Kate?
Well, just because Australia's done better. So we have not had any breakthroughs of patients getting the vaccine and then getting an infection. So I'm sure it's happened, but none of my patients have reported it. So I don't have a good answer for you.
Okay, may I read the questions? Yeah, okay, for Professor, first, Guilhermo, how much time after the COVID-19 they measured antibodies 3 months, 6 months, once the antibodies after COVID go down in a certain time. And the other question is how much time after COVID patients who had COVID-19 received the vaccine.
Bien, lo que hemos visto es que, bueno, además se ha publicado que los anticuerpos pueden durar hasta 6 u 8 meses y si desaparecen...
Antibodies can last 6 to 8 months, and if they disappear, that doesn't mean that it's not immunity. They persist even when they are not antibodies. What we have seen is that individuals that maybe suffer COVID 8 months before getting the vaccine, they showed the effect of intensification with the first dose, which shows that the lymphocytes B and lymphocytes T of memory are being activated. And a few patients who have suffered COVID and did not have antibodies, we saw the same effect with the first dose, which reinforces the concept that with the first dose, what is being reactivating is the memory. And the other question, what was it?
When they gave COVID-19 vaccines in persons who had already COVID.
Sí, es eso. En los que ya tuvieron...
Yes, that's exactly it. Where the ones that already had COVID, up to 6 months, we have seen if they had COVID 6 months before the vaccine, the effect of the first dose is maintained, reactivates the memory of those who already had COVID.
The next question is about the China vaccines. Once they are less effective, if people who received those vaccines are going to receive extra doses from another vaccines. What do you think about it?
Yo creo que la combinación de vacunas debería funcionar.
I believe that the combination of vaccines should work. And even an additional dose of the same vaccine, like what has happened with the Sinopharm, I believe, in the Arab states is worked. It reinforced immunity. I think, yes, it should reinforce as long as the immunogenous is the same, the immune memory should be reinforced even if the adjuvant is different.
Give an opinion about this.
We are using this vaccine in Brazil.
Están utilizando esta vacuna en Brasil. Es frecuente, la estamos produciendo también, tenemos la autorización por parte del gobierno chino. Y aquí la eficacia es alrededor del 50%, así que esto explica que 50% es la posibilidad de que les dé COVID, pero funciona muy bien para reducir la mortalidad. Tenemos que recordar que la campaña empezó en enero con esta vacuna, con un objetivo de los profesionales saludables que trabajan en el servicio público y en la gente añosa. Por el momento, estamos inmunizando a las personas de 40 a 50 años de edad, así que la tasa de mortalidad ha caído en las personas mayores que han recibido esta vacuna. Funciona reduciendo la mortalidad. Hay otra pregunta que quizás me puedan ayudar.
Vaccines in patients that receive IVIG and sub-QIG. Anyone?
Catherine, are you asking about safety or efficacy?
I don't know because there's it doesn't mention it. Just
But
recommendation
I
about
think the security, yes.
I don't know, because security, I think Kate spoke about it. Maybe it's the time for immunization between the application, the infusion of IG. I don't know, maybe.
So
maybe
I'm going to take a stab at a different question. Since I'm not totally sure about that one, there actually is another model out there that I didn't present that shows that plasma derived from the US actually has quite high levels of antibody to SARS-CoV-2, both from natural infection and from vaccines. And the predicted level of antibody in immunoglobulin products is predicted to be therapeutic, meaning it will prevent infection this summer. And I know many countries in South and Central America get their plasma from the US or even the products from the US. So we are very close to having protection from immunoglobulin products. I know that wasn't exactly the question, but it's surprising to me how rapidly it has happened.
No, of course, and it's safe to do to vaccine patients receiving IVIG, and they should receive. You already mentioned that. There's Francisco Espinosa is saying that in Mexico the fatality rate is higher because very few testing is done, and only the severe cases are diagnosed. And the same as in Brazil and many other countries in South America, I think. Let me see if there's another question. No, I can go to questions and answer.
What the Kate, the one question is, what is your recommendation about the use of gamma globulin in the patients and the COVID? What are the moments with the patients?
The immunoglobulin products will be protective. It's just a model. Until we have a lot of patients out there in a community where there's still active transmission, it's going to be very hard to know how effective it is. I have been recommending vaccination for my patients because we're not yet at the point where we think antibody in the products is protective. I don't think I will change my mind in the summer when we think the levels are high enough, because I can't be sure. So I'm not sure what others will do. I know some places the vaccines are very precious, and so you might choose not to give them to someone if you think they're protected. But for me, at the moment, I'm still recommending the vaccine.
And would you recommend the vaccine between the doses of, I think maybe that's the question, between the doses, 2 to 3 days before, 2 to 3 days after? I think this is the question.
Katerini, I have heard that a lot. So I do not. I give the vaccine when I possibly can, and I don't adjust it. And I don't think there's any reason to adjust it. I have not, but there are a ton. You might know that the Immune Deficiency Foundation is having their patient meeting, and all the patients want to know the same thing. So I don't adjust. I think there are people who do, but I'm not sure how they come to the way of sequencing it.
Okay.
Yes, the same.
There's a question about cellular response about 1 doses of immunization in persons who had already COVID-19 before getting the vaccine. If someone knows something about this. Maybe Stu had.
Can you repeat that? Sorry, Katerina, can you just say that question again, please?
Okay, if anyone knows something about cellular answer response about after 1 doses of the vaccine in persons who had already COVID-19 before this immunization.
So are you talking specifically about PID patients or just the general population?
No, I think it's general population. Oscar didn't mention PID patients.
I can't talk about cellular responses, but I have seen papers that have shown that vaccinating with a single dose of people who have been infected, naturally infected, really does boost the antibody levels. So I'm not that familiar with the cellular side of things, but the antibody response certainly does. So it's essentially, that's the old-fashioned way of vaccination. You know, the second exposure really boosts your primary exposure. So I presume, arguably, given that there are pretty good correlates between the T cell and the B cell responses, at least as shown in some nice studies that have come out, particularly from Shane Crotty's lab and others in various places, I would suggest that the same amplified response is seen for the cellular immunity in those cases.
Okay, thanks. And that's a question for Guillermo and for Liliana. The vaccines for SARS-CoV-2 with adenoviral platforms, do they generate immunity also to adenovirus infections?
It's a good question. I don't know, but maybe possible. I guess it is. It protects. I don't have the answer actually, but
I don't know.
It may work. Yeah.
And do you think there's a difference about the length of protection generated by different platforms of vaccines?
Actually, we don't know so far, but I think that with the 2-dose vaccines, the protection may be longer than the single doses, but we don't know so far.
And what do the others think?
They see answer because it's in this moment it's difficult to answer this for me.
Continue, I guess.
Hard to know. It's too early, I think, to be absolutely sure. And particularly, it's going to be the variants are going to complicate that data as well. So we just need the time to see what comes from those studies.
Okay.
Yes, we need to study more.
And Agustina also asked about vaccines in Argentina. How are the situation about development of vaccines in Argentina?
Perhaps I mean
in this moment we begin to do 2 vaccines, Sputnik and AstraZeneca, and that is now in finally on June we are going to use our own production.
No. There's a question about
in June we are going to use our own production in this moment. Yeah.
There's a question
in the
about
future, different lab of the concept lab to do new vaccines. The
We can't hear. We can't hear. No te escuchamos bien, Lili.
Se corta, Lili.
Se me corta. Sorry, because I have the problem with the internet. In the in different lab, the concept lab, the research lab, we produce the own Argentina vaccines. Yeah, the San Martin University to do the new vaccines for our country.
And our group as well.
Yeah.
We are developing
AstraZeneca and Sputnik. Well, we share yet to do it. Them, sorry.
And there's a question about the thrombotic mechanism after AstraZeneca vaccine. If it's the same of thrombosis after heparin.
Yes, the patients present the antibodies en contra, el factor de heparina, ¿sí? 4 es el mismo mecanismo.
On heparin factor 4, it's the same mechanism.
los pacientes,
Patients,
cuando hay es una clínica muy clara de
there's a very clear
trombocitopenia,
clinic of thrombocytopenia
que aparece a los 30
that
días.
appears 30 days in.
Los pacientes no deben ser
Patients should not
con plaquetas porque
be
se
treated with
producen
platelets
anticuerpos contra
because
plaquetas.
they produce antibodies against platelets.
Ser tratados con algunos casos
They
con
should be
anticoagulantes.
treated with
Sí.
anticoagulant in some
Y
cases.
obvio, no heparina
Obviously
ni
not
tratamiento con
heparin
plaquetas, salvo
or,
que sea necesario.
you know, except if it's necessary.
Okay, Stuart, there's a question about NK cells response after COVID vaccines.
For me, I know.
Yeah, so that's really interesting. Obviously, there's been a lot of studies on adaptive immunity, T cells and B cells. There's been a lot of work on the innate system in terms of soluble readouts, cytokines, biomarkers, and so on. But the specific role of NK cells hasn't really been addressed, to my knowledge. Of course, I'm not pretending I'm an expert on anything, but I haven't seen any strong literature showing what the response of the NK cells is like, at least during acute or convalescent responses. So that would be certainly an interesting area to study. We know there's the phenomena of memory in NK cells, more so for herpes viruses, not necessarily for coronaviruses. But it would be interesting to see what the functionality of NK cells is like in individuals who have had asymptomatic, mild, severe, hospitalization with COVID, and see if the NK cells have what they may or may not be doing in that context. But it's a bit of a undersubscribed area of research, I think, from the immunological perspective.
Thank you. And one final question for Kate. Do you measure autoantibodies against type 1 interferon in convalescent plasma for treatment? From Sara Espinosa.
So we have not been, largely because the turnaround time to get the results back is really long. So the assay is only done in one place in the United States, as far as I know, and they freeze the samples and run them. So it's very hard to use that information in real time because it can be a month before you get the results. So we have not been, but there's a lot of interest in, now that this is known to be a part of susceptibility to severe COVID, and because it strikes older men that otherwise don't have symptoms, there's a lot of interest in developing a commercial assay that would be more useful because you'd get results back quickly. So I think it may come because of the pandemic and because it could be used to direct your therapeutics in a more targeted way.
Well, we
Okay.
finished.
I think it's more than 2 hours, so I thank you very much. And Lily, please.
Well, thank you for your generous time. It's a very long webinar, sorry, but it's very interesting and you are very generous with us. Thank you. It's the honor to listen to you. Thank you. Gracias, Ricardo. Gracias, Antonio.
Thank you, everybody. Thank you, Ricardo and Antonio.
Adios.
Pleasure.
Adios, exactly.